So thank you all so much for coming. We had a really wonderful 2 weeks with the Women of Water. Lots of adventures, lots of research, lots of different projects. So I'm gonna give it over to them. Lauren's gonna start us out and we'll just run through these projects. And I'm gonna record it on Zoom, Lauren, so you can.
Record it on Zoom?
Yeah.
Okay.
It is great.
Whenever you're ready.
So as we all know, the water quality is slowly going down in the Gulf of Maine. And one way we can help fight that is through natural filtration with scallop farms and different types of shellfish. So what's the objective? Basically, we're trying to filter out bodies of oceanic water and like tackle harmful like algae blooms. And it's just so it improves water quality and the overall state of life on the bottom floor. So where could it happen? Literally anywhere there is high nutrients and where the shellfish thrive and it's their natural habitat. So this is like a little bit of a diagram of what I'm talking about. So when there's harmful algae blooms, it blocks light from going down to the ocean floor, which decreases the life down at the bottom and this simply makes it kind of a wasteland. By using shellfish farms, we can filter the harmful algae through the nets, through the organic and that's the biology of the shellfish filter the algae through. That's like what they eat. So that's a natural food source for them. And they'll clean it out and we'll bring it so the the numbers of algae down and help like reach the bottom floor to increase life. So basically when there's high nutrients in a body of water, often right by a port or a bay where a lot of pollution's having a lot of runoff, the toxic algae blooms like plankton just kind of explode and that's like what red tide is and that's what and it's not just specifically that one. But the rise in plankton creates an algae bloom which kind of coats the surface so no light can go under. But we can fight that with oysters, shellfish, and any other bivalves that we can put in nets and kind of have it. That's it. I don't know what we're
I'm Maxine Bradmiller.
I'm Lily Hirschson. And our project has explored the amount of microplastics in Penobscot Bay.
So a little bit of context for microplastics and microplastics are considered they are 5 millimeters and under. They're sinking to the bottom of the ocean, which because of processes like biofouling and marine aggregate incorporation, and benthic habitats or any habitats under the water have a higher susceptibility to microplastic accumulation in areas that are known for commercial fishing and lobstering. And there was an increase in microplastic pollution in the ocean.
And so the process of gathering our sediment, we went out on the lobstering boat Sunny and did 3 days of sampling. And we used the Ekman dredge and dropped it to the bottom to gather sediment, but we only have 3 sites because it's fires, sometimes it doesn't close all the way, we lose the sediment. It's a bit of a finicky tool, so it's not perfect every time. But we did get a good amount, and so when we went on our pulling boat expedition, we left our sediment out on trays to dry overnight. And then once we got back, we were probably doing this for like 3 or 4 hours. We measured 1 gram of sediment 3 times from each location, and then sorted through it with the microscope, just counting microplastics. And so the most common microplastics that we found were glitters, microbeads, and microfibers. The microfibers can come from clothes, buoys, and then so for site 1 west of Greens Island, we were going like up the coast of around Hurricane and Vinyl Haven. This is on average, with a depth of 11 meters, there are 55.66 microbeads, 23 microfibers, and 4 glitters. But even though this is an average, and that still is quite alarming, I think it's important to point out that in trial 3 for west of Greens Island, we found 110 microbeads. And then for east of Ledbetter, which was 9 meters deep, on average, we found 16.66 microbeads, 82.66 microfibers, and 10.66 glitters. But again, that's on average, and it is important to point out that for trial 2, we found 122 microfibers in 1 gram of sediment, and then on trial 3, we found 95 microbeads in 1 gram of sediment.
And for site 3, which was south of Neville Island, we found on average 26.6 microbeads, 28.33 microfibers, and around 4 glitters and 1 gram of sediment. Depth was 9 meters.
And of course, there's room for human error. I mean, for 2 people counting microplastics, like it's pretty tedious. You're doing it for probably 4 hours. And so there could be some we didn't count, we missed, or ones that we mistaken for gravel or rocks. And it may not have been 1 gram each time. And there could have also been excess microplastics coming from like skin contact, like coming in contact with the sediment because we were touching it. And then there could have also been microplastics already in the petri dishes, but still, that's like besides the point.
So why does this matter? This matters because the problem with microplastics has only recently reached like the public's mind. Microplastics are very common now in the ocean and everywhere in the world, specifically in the Penobscot Bay. And if it isn't already known, Penobscot Bay is a very prominent area for lobstering and commercial fishing. And as I said earlier, habitats where commercial fishing and lobstering are popular, microplastics are more prominent there. So this matters because microplastics can get into the food we're eating, which can cause scientists theorize DNA damage, inflammation, much more. It could also cause problems for the human habitat. Any questions?
On a similar.
Yeah, on a similar note. Very
Emerson
similar note.
focused on glitter.
I focused on the problem with glitter. My name's Emerson. Nice to meet you all. Anyways, so as the last group told you, microplastics are fragments of plastics that are less than 5 millimeters in diameter. There are 2 types of microplastics. There are primary and secondary. And primary microplastics are microplastics that are designed to be small, such as microbeads or glitter or plastic pellets. And secondary microplastics are fragments of larger plastic objects that have broken apart. And glitter falls under the primary microplastic. And glitter is made by taking a polyester core, coating it in aluminum, and then coating it again in a plastic outer layer. And as you can tell by that list of components, they do not break down easily. It takes about as long as any other plastic object. And the contributors to microplastics are very varied and include textiles, tires, city dust, road markings, marine coatings, personal care, which is where glitter falls, and plastic pellets. And the environmental impact of microplastics is very large from what we can tell. It can prevent certain water plants from being able to grow and take root. It can get into the digestive systems of many different animals, and even into people's bloodstreams who eat animals. And there have been like studies to try and figure out how to get these microplastics out, but nobody can find like a surefire way to get these small particles out of our waters and earth. And the glitter might not be the biggest problem that we have. One thing that I noticed while I was doing my research is that it's one of the only things that falls into the many different contributors that doesn't have an actual like purpose. It's just a novelty. In glitter, we just have it because it's fun. So like we need to wear clothes, we need to have tires on our cars. But there are many options that people can take to use glitters that will not affect the environment so badly, such as cellulose glitters, which I actually used on my poster. Thank you, Kyle, for lending me some. And there's also quinine crystals, which are derived from the scales of fish. And both of these things are naturally occurring cellulose and quinine crystals and will degrade in a matter of a couple months. So in conclusion, there are many different ways that we can prevent like our unnecessary want for like things that are shiny to, it doesn't have to hurt the environment and we can still have our glitter.
Hi, my name's Jillian and I did my project on plankton, and basically I just looked at a lot of the data we found from our plankton samples and a little bit about how we got our plankton samples. We had this mechanism called a Liskin bottle, and it's basically just this very large plastic bottle that you drop in the ocean, and it had these two little caps, and you could like send down this weight to float them. And then to find the plankton, we got some samples of that water from like a little plankton filter, and we looked on them under a microscope while we were on the boat. So it was definitely an experience. But from what we found from our sites, the species that we found, there wasn't that much variation in our species. And as you can see from this lovely chart here, it was pretty similar in the amount of species we found at each site. Though this might not be entirely accurate because you can't find every single plankton in water samples, and also none of us have really ever done this before, so we don't know what's plankton and what's just seaweed or debris. But there were no significant water changes, so as you can see here, there wasn't a lot of significant plankton species differences. So some of the most common species we found are the Pseudomyxia, the Skeletonema fossicum, Ceratium lineatum, there are a lot of big words, Cocinodiscus, and the Pleurocidum. And a little bit about the top three most common ones, starting with the Pseudomyxia. These ones are actually very abundant in our samples, and they're everywhere. They're in each and every one of our sites. And this is an issue because they are toxic, and they create this acid, I think it's like domoic acid, which can build up in the filter feeders that eat them, like scallops and other like filter feeders. And then when we eat them, it causes this syndrome called amnesic shellfish poisoning, which can be lethal and is very bad for people. So that's obviously an issue because they're all around the Gulf. And yeah, so this is also an issue because how harmful algal blooms, like the ones that contain Pseudomyxia, happen normally in warmer waters, which can signify to us that these warmer waters are from climate change, and obviously that's quite a large issue. And then this also impacts the ecosystem by killing off some of the mammals that are eating these, which can obviously cause some very big ruptures in the food chain. And other harmful algal blooms can affect the cleanliness of the water and like the cleanness, and it can impact like different marine mammals' ability to find food, which also disrupts the ecosystem. And even the Pseudomyxia blooms have closed certain fisheries along the west coast. I believe the ones that were the razor clam fishery and the Dungeness crab fishery were closed for a period of time because of the Pseudomyxia blooms, which is obviously not what we want for the economy. And then for the Skeletoninos, they aren't as harmful as the Suganitia, but they are a cause for red tide, and they have caused red tide along the east coast of Asia. And they're also commonly used in some sorts of biofuel because they're very efficient photosynthesizers, and they are very tolerant to pH, salinity, and temperature changes. And we didn't see as much of these as we did with Suganitia, which is probably because they tend to bloom in 25 degrees Celsius waters, but all our samples were around 15 to 16 degrees Celsius. And they're also more common in brackish types of water. And the last one, the Serratia is not as harmful as the other ones, and it doesn't have as much going on. It's kind of just part of the base of the food web, and it photosynthesizes. And it's just a source of nutrients for larger animals and just other plankton. And these are all phytoplankton, which means they're all plankton, but some of the zooplankton we found, and the zooplankton were much less abundant. But we found copepods, Neomyces, cucumber or sea cucumber eggs, krill, fish eggs, and the worm larva thing. And so you might be wondering, why does this actually matter? But plankton are very, very vital to our well-being and the ocean's well-being. For us, they make around 50% of our oxygen. But for the general ecosystem, they create the base of the food web, and they're kind of the bottom, so everything like higher up can have food. Also, the different harmful algal blooms can signify changes in the environment, which generally is caused by us and is something that we need to fix. And these can be like a sign that it's getting worse. And then plankton, like the knowledge of which toxic plankton are in the area is very essential for different fisheries because like the scallops that are grown on the island, you can't necessarily, you can only eat one part of the scallop if they have been contaminated by Suganitia, and for other species, we can't eat any of that because they're toxic to us. And then also, we would want to know what's there to maintain the quality of the waters we're swimming in and getting stuff from. And there are some ways to combat the algal blooms. Some are aeration, chemical and biological additives, and just prevention in general, because if we don't let the waters get this hot, then they won't happen in general. And then we should also be worried about this because the main reason that plankton are being harmed is because of us and because of climate change. So we need to be very aware of how we can keep these very vital species.
So our project is about how do humans use different organisms from the tidal zone. And we came up with this, we focused on two of them, Irish sea moss and green crabs. But one day this week, we went down to the intertidal zone and collected both Irish sea moss and green crabs to use in different ways that will get more.
So Irish sea moss is a type of seaweed that grows in the low intertidal zone. It is used to make pure gluten, which is why we use it as a clean agent, mostly in dairy products such as ice cream, chocolate milk. It is also used in the toothpaste. It can also be used in meats, deli meats. So, it's a replacement for gelatin with vegans and vegetarians. Yeah, we used it this week. We cooked it. And we put, I think we have like a bucket full of Irish sea moss, and we boiled it until all the pure gluten was out, and then we strained it through a strainer. Couple of shrooms. And then we added a bunch of ingredients such as like chocolate, cinnamon, maple syrup, sugar, and made a pudding out of it, and blended it and made pudding.
Yeah,
How was it?
it was good.
It's interesting.
Yeah.
It was like a 2-bite.
Yeah.
dessert.
Yeah.
It
It was the best seaweed pudding I've had.
She dumped like a whole thing of sugar in there.
And it was good. It was good.
Yeah, and then we also have green crabs. Green crabs are an invasive species and they are very harmful to our ecosystem. They eat clams, oysters, mussels, and other small crustaceans. And they're especially detrimental to the soft shell clams. And they're very resilient, and they have adapted very well into this new environment. But humans have found a couple of ways to use them in a positive manner, and one of the main ways is that they eat them. There's a bunch of different ways that you can cook them, and in Italy, especially in Venice, they're a very popular food. People use them for, they're harder to row in some places. It's often sauteed with pimento pasta, and we want some recipes. They can be eaten as pate. Some people also boil them and they use them as stock. Some of the larger green crabs can be shucked, and they can also be fermented, and it's very common to at the replacement for other small crabs in Cambodian, Korean, and Thai cuisine. Soft shell crabs are also popular in some places. The thing that we did with the green crabs is that we made bait. And you can buy special green crab traps to catch the green crabs. And then it is using green crabs helps control the problem with green crabs. It also helps the fishing of common bait fishes. But the way that we made bait is that we collected a bunch of green crabs and then we blended them. And we put some agar in and boiled them. And then it created like a solid kind of jelly-like substance. And then we went lobstering the next day and we were able to cook them in the lobster.
That concludes our presentations for the Women of Water. Thank you all so much for coming. We had a really wonderful 2 weeks, and hopefully you learned a little something, new stuff that they learned.